Good day, Prof. Shahil and Dr. Huzalina. We are from Group 12, presenting the assignment for Biocomputation and Bioinformatics. Our group member consists of Li Zishen, Chao Wei-wen, Mao Mitamura, and me, Leong Chi Kei. Now I will continue on the introduction. The enzyme that we choose to use in this assignment is beta-glucosidase with EC 3.2.1.21. It belongs to glycoside hydrolase families. Beta-glucosidase can hydrolyze the beta-glycosidic bonds in complex cellular structure into beta-glucose. Secondly, it also helps in lignification and catabolism of cell walls in plant and biomass conversion in microorganisms. When beta-glucosidase is in human, it can help to catalyze the reaction of glycosylceramide to transglycosylation to form cholesterol. Secondly, in biomedical related research in the metabolism of lipid steroid and sphingolipids in terms of health and disease in humans by using beta-glucosidase. Dermatoga neapolitana is a gram-negative bacteria. It can hydrolyze the nickname cellulosic waste like hemicellulose by breaking down the beta-glycosidic bonds at the non-reducing end of glycoside.
Thank you, Chike, for the introduction. Now, I am Li Zishen. I will explain the methodology. As mentioned in introduction, our group choose beta-glucosidase as the enzyme, and we will choose two different organisms to compare the different sequence among these organisms in beta-glucosidase. So our group choose human and also thermobacteria. So in human, we are using the Homo sapien Q9H227, and in thermobacteria, we are using Dermatoga neapolitana B9K7 and 5. So both of them, we are using the Uniprot database as the database gene to know the sequence of the protein. When we know the sequence of these two enzymes, we will do the sequence alignment by using T-Coffee and also Uniprot. Once we do the sequence alignment, next we go to the visualize the 3D structure of these two enzymes. So we visualize the 3D structure based on the database from RCSB PDB. So when we copy the accession number from the Uniprot to PDB, for human, we will get the 2E9L accession number. And for the bacteria, we will get the 5IDI accession number. And under the same website, same database, we will also do the multiple pairwise alignment under these two 3D structure of this enzyme to see whether the active site, binding site, and also other property of the enzyme are similar or not. Next, we will do the phylogenetic analysis. So first, we need to do the alignment between these two enzymes first. Once we get the sequence alignment between these two enzymes, we will compare this the sequence with the bank, with the database bank by using the BLAST to find the best 20 possibility matching enzyme from this alignment lah. So once we get the 20 best alignment, 20 best enzyme, we will do the cluster alignment among these 20 enzymes, and finally we were using the Mega 11 software to construct the phylogenetic tree. Finally, we will do the auto.vina to determine which enzyme has the higher affinity or higher interaction of between active site and the substrate. So in auto.vina, receptor will be our enzyme, and our ligand, which our substrate, we are choosing cellobios with the PDB ID 2BOD. When we want to run auto.vina, we need to do two things first. We need to prepare the receptor and also prepare the ligand using the Autodoc Vena software. Firstly, for the receptor preparation, we need to launch the two enzymes and that's already saved in the PDB format from PDB database will be launched in the DS visualizer to remove all the water molecule in the enzyme. And then after that, we will launch these two enzymes again using the Autodoc Vena software to color the atom types and also adding the hydrogen atom. Next, we will assign the charge and also atom type to the enzyme molecule, and then this enzyme will be saved into the PDB Qt format for the Autodoc Vena running. And finally, we need to define the 3D space so that to let the computer software know that which location will be the active site. So later I will show the diagram where for these two enzymes where the location of the active site are. This slide shows how we define the 3D space to locate the active site of the human enzyme. So the left-hand side is the information about the good box, the location, the x-axis, y-axis, and z-axis of the grid box, and the right-hand side is the actual location, the 3D location of the active site. And this slide is show the define the 3D location of the thermobacterial enzyme. So you can see the left hand side is the information about the location of the grid box, and then the right hand side is the actual location, where is the active site found and where is the 3D space where the substrate will be bind. And then once we already prepare the receptor, now we need to prepare the ligand. So normally, first two steps are similar to the receptor preparation, which is launched in the DF visualizer to remove water molecule, and then launch in Autodoc Vena to color the atom type and also add hydrogen atom. And finally, we will define the ligand in the Autodoc Vena software. And once we already do the ligand preparation and also receptor preparation, we can start to run the Autodoc Vena, where the interaction will be occur using the Autodoc Vena software, and you can visualize the result using the Autodoc Vena and also PyMo. And the result and discussion will be discussed by another group member. So that's all for my part. Thank you.
So next is about the result and discussion part. This is the sequences of our enzyme in FASTA format. So left side is the sequences for Homo sapiens Q9H227, while right side is the sequences for Dermatoga neapolitana B9K7M5. This is the amino acid sequence analysis. The total amino acid length for Homo sapiens Q9H227 is 469. It has two active sites at which both involve glutamic acid. It also has 6 binding sites which involve glutamine, histidine, asparagine, tyrosine, and tryptophan. For Dermatoga neapolitana B9K7M5, it has total amino acid of 444. It has 2 active sites at which both are also involve glutamic acid. It has 7 binding sites which involve glutamine, serine, histidine, and glutamic acid. The sequence of the active site of both enzymes are glutamic acid. Here are the sequence alignment results from T-Coffee and UniProt. In T-Coffee result, the red color region indicates good alignment, while the yellow and green regions are not well aligned between the sequence. The T-Coffee alignment sequence gives 812, which indicates the significant reassemble level between the two sequences. In UniProt result, the asterisk symbol gives the meaning of identical amino acid sequence. Colon symbol means similar amino acid sequence but not identical. And the dash symbol is a gap that introduced to make the overall length of the alignment same. So the percentage identity metric of these sequences generated from UniProt is 40.23%. This indicates the moderate similarity between the sequences. Next is about the 3D structure. These are the 3D structure of our enzymes downloaded from PDB. Secondary structure analysis had been done. The Homo sapiens Q9H227 structure consists of 20 helix and 16 sheet structure, whereas for the Dermatogania pelitana B9K7 and 5, the structure consists of 23 helix and also 15 sheet structures. This is the 3D structure alignment result of the two enzymes. Blue color is the structure of Dermatogania pelitana B9K7 and 5. Brown color is the Homo sapiens Q9H227, while the purple color is the active site. This 3D structure alignment results has shown that the active site of the both enzymes are located at a similar location.
Now let's discuss about the phylogeny. These are the reasons why we do phylogenetics analysis. One is to determine the closest relatives of the organisms. And the second one is to discovering the functions of a gene. And the third one is to retracing the origin of a gene. We use Mega 11 to construct phylogenetic tree. In Mega 11, we can not only construct phylogenetic tree, but also can compare sequences. By comparing sequences, using alignment of Homo sapiens and Dermatoga neapolitana is compared to other sequences in gene bank using BLAST to determine the most 20 possible similar enzymes, and then align all sequences by cluster W. After that, we can construct UPGMA tree with 1,000 bootstrap cycle. This diagram shows the phylogenetic tree of the 20 most possible amino acid sequences in different organisms by using the cluster W alignment sequences between Homo sapiens and Dermatoga neapolitana. The phylogenetic tree is composed of branches and nodes. The branch is defined as links between the taxa in terms of ancestry or the tree's topology, and one branch will connect to any two nodes, which is the intersection point of the branches. The branch that forms towards the operational taxonomic unit is known as the external branch, while the others will be known as the internal branch. It is shown that there are 20 operational taxonomic units and 19 internal nodes in our phylogenetic tree. Roots in the phylogenetic tree represent the most recent common ancestor of all the sequences, while our phylogenetic tree shows it is rooted at the bottom and divided into two branches. The branches show how these descendants are related to each other with the common ancestor. A branch's length indicates how many generations have gone by since a particular lineage split off from the main family line. Our phylogenetic tree shows that the bottom branch from the root has a longer length compared to the top branch. This shows that the beta-glucosidase sequence for the top branch shares many common ancestors. Branch length is very important in the tree, where it shows how many amino acids are changed in the branch. The longer the branch length, the higher the change of amino acid sequences from the initial ancestor. This format is called a phylogram that gives a clear visual representation of how connected certain proteins are to one another within the tree. Based on our phylogenetic tree, the top lactase fluorescein hydrolase of Ovis aries shows the shortest branch chain, which shows that amino acid sequences have minimal evolutionary divergence. Full beta-glucosidase of Calycellulosiropter saccharolyticus, Paenibacillus polymyxa, and Nealia succulans, beta-cloto of Mus musculus and Homo sapiens, and cloto contains Lecnem of Lactus norvegicus, Mus musculus, Homo sapiens, and Macaca fascicularis show the longest branch lengths, where there is significant evolutionary divergence among them.
Hi, I'm Chiki. I will continue on the orthodox vena part. From this slide is shown two figures. The first figure is the binding affinity between 2E9L and 2BOD, while the second figure is the binding affinity between 5IDI and 2BOD. For this slide is about protein-ligand interaction. The ligand that we choose for this assignment is cellobiose with PDB ID 2BOD. Cellobiose is a disaccharide consists of two glucose linked by beta-glycosidic bond. From the previous slide, we have shown that the highest binding affinity between Homo sapien and cellobiose is achieved at negative 7.7, while for the highest binding affinity between Tomotoga and cellobiose is achieved at negative 6.9. This can conclude that there is a stronger binding affinity between Homo sapien and cellobiose because the lower the affinity value, the better the binding affinity. This is the interaction between enzyme and ligand present in orthodox. The part circled in red indicate the active site for each enzyme. We have visualized the enzyme and ligand in blind mode. The left figure is Homo sapien and cellobiose, while the left figure is Tomotoga and cellobiose.
This is our conclusion part. In conclusion, the comparative analysis of beta-glucosidases between Homo sapiens and Tomotoga neopolitana reveals significant differences in enzyme sequences, structures, and functions. Understanding the differences in active site locations, amino acid sequences, and structural features of beta-glucosidases enhances our knowledge of how these enzymes catalyze reactions and interact with substrates. Overall, the comparative analysis provides a foundation for future studies, exploring the roles of beta-glucosidases in health, diseases, and metabolic processes, offering insights into the intricate mechanisms underlying enzyme functions in different species. Thank you so much for listening.